Hello, my name is Shyh-Dar Li. I'm Associate Professor at UBC Faculty of Pharmaceutical Sciences. My research focuses on drug delivery technologies. Today, I want to talk about one particular drug delivery system or nanoparticles we have been using for drug targeting, that is liposomes. So in this slide, you can see that liposomes, we can view liposomes as a nano-sized cell with a lipid bilayer. And we can use this structure to deliver a variety of drugs, including hydrophobic and hydrophilic drugs. For hydrophilic drugs, we can put them in the inner aqueous core, as I pointed out in the slide. For hydrophobic drugs, we can put them in the lipid bilayer. But there's a significant problem with delivery of hydrophobic drugs. As I mentioned, normally they are encapsulated in the lipid bilayer, but there are two limitations. The first one is the lipid bilayer capacity is very low, so the loading efficiency is low. Second, the drug association with the lipid particles is poor. So that means when you inject the formulation into the blood circulation, the drug tends to leak out, and then you don't have the advantage of using liposomes to deliver anymore. So here is an example. So in this case, docetaxel, which is a taxane drug, very hydrophobic, is loaded in the liposomes. So in this case, in the closed circle. And also in this figure is looking at the pharmacokinetics after IV administration in comparison with a formulation that is containing a detergent and a solvent called ethanol. So the formulation is called Taxotere, is used clinically. And in this case, you see that the blood concentration and time profiles for both formulations are similar. They are overlapping. So there's no clear advantage of using liposomes in this case compared to the standard formulation Taxotere. Again, that's because the drug and nanoparticle association is poor. And that's why people develop different approaches to improve the drug loading into liposomes. So in this case, we call active loading. And active loading is normally developed or used for amphipathic drugs, and you can see the reason when I explain the active loading. So this is a liposome, and then we create the liposome containing a pH gradient. Inside the pH is about 4 and outside is about 7. And in this case, when we need to load a weak-based drug, when the drug is dissolved in the higher pH conditions, and you see the drug is mostly in the free form, non-ionized form. So it's membrane permeable and can get into the aqueous core of the liposomes. But once it gets into this low pH environment, it will get protonated and then locked in the liposomes because charged molecules is no longer membrane permeable. So this method is called active loading. It's very efficient because we prepare the liposomes containing a gradient first, and then all we have to do is mix with the drug, and the loading will happen by itself with a very high efficiency and percentage. So here is the example of Doxil, which is the pegylated liposomal doxorubicin used clinically. And this is under the cryo-TM. You can see the morphology of Doxil showing lipid bilayer, and the drug is now precipitated in the aqueous core. So again, with this type of loading technology, the loading efficiency is much improved, and with also stronger association between the drug molecules and the lipid nanoparticles. So this is just a proof of the strong association of the particles and the drug, leading to improved pharmacokinetics and the tumor delivery. So this is looking at the tumor uptake of drugs, the drug doxorubicin delivered in the free form or in the doxil formulation. And you see that without any formulation, the drug is the drug delivery to the tumor is quite poor. But with the doxil formulation, with the active loading technology, the curve is much higher, and also the area under the curve AUC is increased by 30-fold compared to the free drug formulation. So this just shows you the how powerful this active loading technology is to improve drug delivery. But again, as you probably can appreciate, there are some limitations for drugs that can be actively loaded into liposomes. First, they have to be membrane permeable, and they also have to be water soluble. So that is not the case for hydrophobic drugs that we are hoping to improve the delivery for. So in my lab, we develop a different version of active loading technology called solvent-assisted active loading technology, abbreviated as SALT, for poorly water-soluble drugs. Okay, so this is a diagram of the SALT technology. So again, we have liposomes containing a gradient, and in this case, we put a trapping agent inside the aqueous core, but outside we don't have this trapping agent. So again, it's just to create a gradient driving the drug into the aqueous core. But different from the standard active loading technology is that we incorporate a limited amount of water-miscible solvent, such as ethanol, DMSO, or THF, that type of solvent. So the reason that we include a limited amount of water-miscible solvent is that we want to help insoluble drugs to dissolve in this mixture, but in the meantime also improve the membrane permeability to allow drug molecules to penetrate easily. Okay, so now the drug is separated as individual molecules, and then they can penetrate into the aqueous core and then interact with the trapping agent inside. And at the end, we perform dialysis to remove the solvent and also unencapsulated drugs to get the final formulation. So I'm going to introduce 3 examples that we use this technology to generate. And so the first model drug is called Starsporin. It's a tool compound in medicinal chemistry because it's a pan-tyrosine kinase inhibitor, very potent, but also very toxic because it has no selectivity. So in this case, we just want to demonstrate that our technology can work for this compound. So you can see the structure of Starsporin here. So you probably can appreciate that it's a very, very hydrophobic drug. The water solubility is less than 5 microgram per ml. But the good thing is that it has one ionizable group here that we can utilize to trap the drug inside the liposomes using pH gradient. So we generate the ammonium sulfate gradient liposomes and then compared the loading efficiency of the Starsporin into the liposomes in the presence or absence of a DMSO. So in this case, we are comparing no DMSO in the loading mixture or the loading mixture containing 5% DMSO. You see the huge difference that without DMSO, there's no loading. because the drug precipitates outside the liposomes, and the drug molecules are not able to penetrate into the aqueous core. But with 5% of DMSO, we could achieve 100% encapsulation efficiency. And I just want to show you how they look under the electron microscope. So again, this is a liposome, and you can appreciate that this is a lipid bilayer here, and the loading capacity is pretty low in the bilayer. But in this case, we indeed load Starsporin into the aqueous core of liposomes, and which you can also appreciate the loading capacity is much larger in the aqueous core compared to the lipid bilayer, and Starsporin is forming aggregates inside the liposomes. So our technology is working with weak-based drug. How about weak acid drug? So that's why we introduced the second model drugs, gambagic acid. So gambagic acid is an active ingredient in the Chinese medicine. So later on, it was found it has many different activities, including anti-inflammation, anti-angiogenesis, and anti-proliferation. So it has been under development for anti-cancer drugs for a while. But the major problem is that water is soluble, and also it has pretty high toxicity. So again, our approach is that can we formulate this drug into liposomes, and then so that we can increase the solubility, but in the meantime reduce the toxicity by increasing the tissue targeting. So again, we use the salt technique and then load gambagic acid into the liposomes. So you can see that we still can load a weak-based drug like gambagic acid into the liposomes. So this is lipo-GA compared to empty liposomes. You see that there's a very interesting bilamellar structure here, and you see the electron dense structure inside. Again, suggesting that during the loading there's some lipid rearrangement and the drug is loaded in the aqueous core. So as I mentioned, the drug has pretty high toxicity, especially during the IV injection. It really causes a lot of discomfort to patients. And also one of the indicator is hemolysis during the infusion. So a very significant problem. So our first study with the lipo-GA is to see that with the liposomal encapsulation, can we reduce the hemolytic toxicity of gambagic acid? So in this case, we have arginine, which is the solubilizing vehicle for gambagic acid in the clinical trials. And we have free GA, that is GA dissolved in the arginine-based water vehicle. And we have empty liposome GA at low concentration, and liposome GA at the high concentration. Again, you can appreciate that with the free drug is really toxic, inducing significant hemolysis. But once we formulate the drug into our liposomes, you don't see this kind of toxicity anymore. As I mentioned earlier, the goal is to produce a formulation that will have a strong association between the drug molecule and the lipid nanoparticles, so that we can really prolong the pharmacokinetics and improve the drug delivery. And as you can see here, this is looking at the blood concentration over time after IV injection, and we compare two different formulations. One is the free GA dissolved in arginine-based based vehicle, and another is lipo-GA. So the square is lipo-GA, and you can see the blood concentration is much higher compared to free GA, and the half-life is increased by